Hello friends, this class we are going to have discussions on fertilization in angiosperms. From the previous knowledge, we have learnt different aspects of plant reproduction, especially on floral structure, development of male gametophyte and female gametophyte, where the male gametes and female gametes are present. The next event is fertilization, and one important aspect is already discussed, that is pollination, the mechanism by which pollens land on the stigma of the flower. But fertilization is very important in the reproduction, because fertilization leads to formation of seed and subsequently fruits. Bryophytes and pteridophytes, they are the amphibians of the plant kingdom. Why? Now, because they need water for fertilization, especially the antherozoids, they need a water film to swim from the antheridium to the egg present in archegonium. Therefore, they need a water film, but when we come up in the evolution, we have seed plants, spermatophytes, the gymnosperm and angiosperms. They do not have swimming sperms, they have non-motile sperms. If you look at fertilization, and here we see the different events of fertilization, but let us come to the discussion, that is how the seed plants require a pollen tube for carrying the sperms from the pollen to the egg present in the ovule. So, now in the seed plants, the sperms are non-motile, they cannot go by their own, they must be carried by certain structures, and these are, that is the pollen tube. If you look at the male gametophyte, that is pollen grain, it lands on the stigma, but the female gamete present in the ovule, it is deep seated in the ovarian cavity. So, how the sperm, non-motile sperm can travel to the egg present in the ovarian cavity. So, that is why it has to cover a long journey, and the pollen grain has all its structures to do so. Let us see, in gymnosperms, the pollen grains take lot of time to start their journey from stigma to the ovule, but in angiosperms the pollen tubes are fast growing. It is an evolutionary advancement over gymnosperms. In gymnosperms, pollination and fertilization, there is a big gap, it may be months or years or so, but in angiosperms pollination is quickly followed by fertilization, as pollen tube very soon germinates on the stigma and sends a pollen tube that reaches the ovule, bringing about fertilization. Now, but pollination is a random event, pollens may be carried by the wind or animals, insects, whatever the means may be, but it is a random event. Any pollen may land on a stigma of a species, but thing is it a right kind of pollen? May not be. It may be right kind of pollen or wrong kind of pollen, but only the right kind of pollen has the capacity to go through the style of the pistil. But how the plant knows which pollen is right kind or wrong kind? So, for that reason the pistil has the ability to recognize the right kind of pollen. If it is wrong kind of pollen, then it can be rejected. It will not be successful in sending its pollen tube towards the ovule. So, now in this context it can be told compatible and incompatible pollens. right kind of pollens can be said to be compatible and wrong types said to be incompatible. Now, a pollen, as we have learned, is surrounded by exine, the outer wall. Besides this or outer to this exine, it has a pollen kit or sometimes called pollen coat. It has lipids, carotenoids, many compounds in this, and it is learned that this pollen coat, which is very vital in recognition of or it is recognized by the stigma. A pistil rejects an incompatible pollen either by preventing its germination or abortion of the pollen tube on the way. Rejection can be at any moment. Some may not be allowed to germinate on the stigma, and others, those who have germinated and penetrated to certain extent in the style, they are on the way they are aborted. So, in words we can say this, but this pollen and pistil interaction is somewhat chemical. Their recognition, identification, rejection, everything can be described in terms of chemical dialogue, but it is not easy to define this complex system. I will tell you in one instant how a pollen tube is rejected or aborted on the way, the mechanism. Okay. Now, we have knowledge that many pollens are shed at two-celled stage. Almost 75% of the pollens are shed from the anthers at two-celled stage, and around rest at the three-celled stage. Those which are released at two-celled stage, the pollens or the male gametophytes, they complete their rest of the development on the stigmatic surface. There, the generative cell divides mitotically, resulting the two sperms, non-motile sperms. Now, the two sperms are present or floating in the general cytoplasm of the vegetative cell. So, this germination of pollen grain is an important event. As we know, pollen grains have germ pores, the weak areas of the exine, where exine is either absent or thinly deposited. In these areas, a pollen tube would emerge, will come out, but for that reason we must know that the pollen grains during the release from the anthers, they are already in almost a kind of a dehydrated state and metabolically less active, but upon hydration, upon landing on the stigmatic surface, they are hydrated by the exudate of the stigma, wet stigma. Wet stigma releases certain liquid rich in sucrose and other things on the surface, and that is absorbed by the pollen and it gets metabolically active. Then only through the germ pore, the intine is pushed out to form a pollen tube, and the pollen tube grows by extension of the tip. It is a very dynamic system. Pollen tube growth is a very dynamic system in the style, right. The pollen tube growth is apical and it is always pushed, in the style because of apical growth due to turgor development or turgor pressure development in the tube, and the cell wall is very thin. The pollen tube wall is very thin, and that is made of pectin and cellulose, and this way the gametophyte pushes in the through the style. But in the picture you can see through fluorescence microscopes, the pollen wall and the pollen tubes, they fluoresce, and these are the structures, the germinating pollens. Now, look at this picture. A pollen tube grows very fast. You can't imagine, it is one of the fastest growing cell. almost in general 10 micrometer per minute it grows in the style, and up to 650 micrometer has been recorded in chicory. This pollen tube growth is very fast one. How this is done? Now, when the pollen tube grows deep in the ovarian in the style, the content of the pollen tube is going down, but from rest of the empty spaces it is plugged because of the development of callose plugs. So, callose plugs are laid down at intervals to prevent backflow of the contents into the back portions. So, now you can see this from this diagram, how these microfilaments or the actin filaments are involved in the extension of the pollen tube, and Golgi derived vesicles, they are the cargoes, they carry the cell wall material to the tip and they are the fuse, and ultimately the pollen tube grows by extension, and at the tip of the pollen tube many vesicles are organized or they are found. It is because these vesicles carry the raw materials for the synthesis of cell wall they are in. Now, this pollen tube growth is the responsibility or the function of the vegetative cell. Therefore, the vegetative cell carries out this extension of this. Now, at this juncture, even after having knowledge of pollen tube growth, let me tell you how a chemical dialogue or interaction between the style and the pollen tube or the male gametophyte results in abortion of the pollen tube. Now, let us see how a pollen tube is inhibited or aborted on the way by the style. Now, this is the stigma and the style traverses through this, and this is the ovary. So, my question is, when a pollen tube grows like this by its tip extension, in case of compatible pollen, how this happens? In compatible pollen or compatible type of pollen, the pollen tube grows, but in case of incompatible pollen, the pollen tube incompatible pollen, the pollen tube is aborted. Pollen tube is aborted. How this happens? So, this style has an RNase called sRNase by stylar RNase. This stylar RNase, this is a protein. This stylar RNase, in case of incompatible pollen, this stylar RNase enters the pollen tube, thereby degrading the rRNA present therein. So, when rRNA is degraded, definitely all its ribosomes are degraded and the pollen tube is defunct and it does not grow, it is aborted there on the way. But in case of compatible pollen, this stylar RNase enters there, but it is degraded by another protein. Therefore, sRNase, even though it enters in case of compatible pollen, it is degraded therein. Therefore, the pollen tube grows. Now, what kind of tissue in the style allows the pollen tube? It is found that a specific kind of nutritious tissue called transmitting tissue. This transmitting tissue provides nutrition to the style, it is compatible to the style, it allows the growth. There are two kinds of styles depending on the structure, one is solid style, the other one is hollow style. The pollen tube grows being lined with the hollow style, but in the solid style it penetrates through the tissue, transmitting tissue. Anyway, now in these two diagrams you can see at many places in these long styles the callose plugs have developed that prevents the backflow of the contents of the pollen tube. Now, the second diagram is that of the Helianthus sunflower, which is a solid style, and the transmitting tissue is shown therein, and this transmitting tissue allows or help the growth of the pollen tube. Now, the pollen tube always grows towards the synergid of the embryo sac. Synergid is a secretory tissue and it provides a cue, that means calcium gradient is created by the synergids. So, that pollen tube is always growing towards the embryo sac. Now, whatever the means may be, but here it may be discussed that the pollen tube enters the ovule in three ways, generally through microphile, which is called porogamy, or through integuments, it is called mesogamy. If it goes through chalaza, it is chalazogamy. The synergids are secretory cells. They secrete calcium, which forms a gradient, and as a result of which pollen tube always exhibits a chemotropic movement, and when the pollen tube reaches through the microphile to the synergids, there it ruptures. The pollen tube ruptures, as a result of which it is released to the synergid, one of the synergid. Now, the fate of the two sperms is to fertilize the egg, the central cell. In the female gametophyte, as we know, there are 7 cells. At the microphilar end, there is your egg apparatus consisting of egg cell and 2 synergids, the central cell, a big cell whose future is to form the endosperm, and 3 antipodals. Now, out of when the pollen tube is released, the pollen tube bursts and the 2 sperms are released, they localize themselves in between the egg and the central cell. One of the sperm fuses with the egg cell, the other one fuses with the central cell. After fusion of the 2 cells, then follows karyogamy. The first fertilization of the egg and the sperm results in a zygote. In the zygote, the 2n nucleus is formed, and the other one, where in the central cell already 2 haploid nuclei are present, and 1 sperm cell fuses with that, and 3 haploid nuclei fuse there, and this phenomenon is called triple fusion. So, as 2 sperms fuse 2 different cells, the first one is the zygote, egg forming zygote, which is called generative fertilization, and the second one, why it is called generative fertilization? Because the zygote ultimately will generate the embryo in the triploid endosperm cell, which is called primary endosperm cell. As it is the first starter cell, it will form the endosperm of the seed, then that is why it is called primary endosperm cell, which is a triploid cell having triploid nucleus, a large cell present in therein. Now, as 2 fertilization events are taking place, this phenomenon is called double fertilization, and in the second cell, as 3 nuclei fuse, that is called triple fusion. Fertilization was discovered by Strasburger in 1884, but the phenomenon of a double fertilization was discovered by Navaschin in 2 genera, one is Fritillaria, another is Lilium. This double fertilization is present in all angiosperms. It is almost unique to angiosperms. The double fertilization has contributed certain advantages to angiosperms, as a result of which angiosperms are more advanced than gymnosperms. So, we will see how the advancement is, because that will lead to formation of seed and fruit. Now, following double fertilization, what are the events? What happens to the 7 cells present in the embryo sac? The 3 antipodals present at the chalazal end, they degenerate. The central cell upon fertilization has been a primary endosperm cell. The 2 synergids already have played their role. They have guided the pollen and facilitated fertilization. So, the synergids degenerate, only 2 cells, the diploid zygote cell and triploid primary endosperm cell, they will undergo further development and the surrounding integuments also. So, now fertilization, an event which would lead to development of endosperm and embryo subsequently. So, the process of endosperm development and embryo development is called post fertilization events. Now, the primary endosperm cell is vacuolated as already mentioned. This divides repeatedly. One important phenomenon in this, the nucleus is very active. This nucleus divides repeatedly by mitosis, but no cytokinesis follows this. As a result of which, in the single cell, due to mitotic divisions, many triploid nuclei are found. So, this cell now is called liquid syncytium. It is a soinocyte. It has in its single cell, it has large number of triploid nuclei. So, this stage is called nuclear stage. What happens next? Let me tell you why this endosperm takes place first, because zygote will give rise to embryo. In the plants, to assure nutrition to the developing embryo, first the plant has activated the endosperm development. This endosperm will provide nutrition to the embryo. That is why endosperm development precedes that of the embryo development. Now, this endosperm is a multinucleated structure, many nuclei are present and this is called liquid syncytium. If you look at the coconut, a young coconut, green coconut, when filled with only water, only milk, that is the liquid syncytium, but subsequently cellularization takes place. Around each nucleus a bit of cytoplasm, then cell wall formation occurs. So, cellularization or free cell formation follows that of the mitotic divisions, as a result of which the milk is converted to meat. You can see in the coconut endosperm there is milk part as well as meat part, but here I can say what triggers, what induces so quick development of the endosperm. Chinese scientists, they were using coconut milk for tissue culture purpose, because this young milk or milk possesses very high concentration of cytokinin, a cell division promoting hormone. Therefore, because of presence of or accumulation of cytokinin in this primary endosperm cell, the nuclear division is hastened or goes quick, very fast. Now, once the endosperm is formed, it is followed by the development of the embryo. Now, you can look at here, the endosperm is rich with many nuclei, but embryo development from that of the zygote by way of mitosis occurs. Now, we cannot say after completion of development of or after development of the endosperm then follows that of the embryo. It is not like that, it goes on synchronously, simultaneously, first endosperm followed by embryo, but they go or they develop side by side. Now, you can look the embryo when the zygote divides, it gives rise to a multicellular structure and there is polarization, it gives rise to embryo proper and a suspensor cell. The suspensor is a transfer cell, it transfers nutrients from the endosperm to the embryo. You can look at there is a basal cell that through that cell the nutrients from the endosperm are diverted to the embryo, so that the embryo grows. Now, embryogenesis is the formation of embryo. So, the initial structure is the proembryo, then it gives rise to a globular structure, then a heart shaped structure, and finally, a mature embryo results forming two cotyledons. So, the different stages of embryo development are proembryo followed by globular stage, heart shaped and mature embryo. The embryo development is so to be synchronous with that of the endosperm. You can look at this now. The embryo at the end, it has differentiated a lot and at the cost of the endosperm. We are going to discuss regarding the post fertilization events in the flower. Post fertilization events leads to formation of seeds and fruits. In the previous discussions, we have or we discussed the development of endosperm and embryo. Endosperm and embryo development goes on synchronously, and embryo of monocots and embryo of dicots in their early embryogeny, in early phases of embryo development follows similar pattern. I told you earlier that there is initially globular stage, then heart shaped stage, then torpedo or mature stage the embryo goes for. So, embryo goes through different stages of development, and but finally, the embryo of monocots and dicots they differ in their structure, and we are acquainted with dicots and monocots. Dicots means dicotyledonous. Dicotyledonous means two cotyledons present in the seed. Let us have a look at the structure of a dicotyledonous seed. Dicots means two cotyledons. So, two cotyledons are present in the seed. So, dicotyledonous seed has one embryonal axis. This axis is your embryonal axis, and to this embryonal axis there are two cotyledons attached, and these cotyledons are storage leaves. They are storage leaves. Their function is to provide nutrition to this embryo during germination. Now, this embryonal axis consists of has two ends. At the top it has plumule or the shoot tip, future shoot tip, and at the lower end it has radicle or the root tip. Now, what should be taken into consideration? The portion or above the point of attachment of the cotyledons, this portion is called epicotyle, and the portion below the attachment of cotyledon is your hypocotyle. This is hypocotyle. We can soak certain seeds like your groundnut or other seeds we can soak, and after swelling or imbibition you can break it and you can see the embryo and cotyledons and the integuments whatever present they are in easily. Now, a typical dicotyledonous embryo consists of one embryonic axis and two storage structures, and this embryonic axis having the plumule which gives rise to the shoot tip and the radicle which gives rise to the root tip. But on the contrary, a monocot embryo is different, but look at this picture. This is that of the groundnut and this is another of the bean seedling. You can easily assess the point of attachment of the cotyledon. In the groundnut, the cotyledon is fleshy, it is the storage structure, and a small plumule having the leaves are seen in this, and the integuments you can see easily, it covers the groundnut, and another is the germinating bean seedling. Here you can easily see the hypocotyle, because these beans exhibit apigeal germination. This hypocotyle grows faster than the epicotyle, as a result of which whenever you have put this in the soil, as the hypocotyle elongates faster than the epicotyle, that is why the cotyledons are pushed above the soil and they become photosynthetic and they support the seedling for growth. On the contrary, monocots have one cotyledon, monocotyledonous or monocots in short, they have one cotyledon, but the cotyledon hardly we see in monocots. Where that cotyledon lies? The monocot seeds like wheat, rice, maize, these are filled with endosperm. You can see from this diagram, the outer is pericarp, inner to that it is mostly filled with endosperm, but scutellum is the cotyledon. It is not fleshy as it is in dicot. Scutellum is a rudimentary tissue, highly reduced, papery structure. It is the interface between the embryo and the endosperm, the food reserve. And the embryo proper has the plumule, the shoot tip, radicle, the root tip, but the shoot tip is protected by coleoptile. Hardly we can imagine what a coleoptile is, unless we germinate this. We have no microscope here, we cannot see this, but upon germination I can show you what a coleoptile is. So, coleoptile is shoot covering, it is an undifferentiated sheath around the plumule. Similarly, above the radicle, the root tip is the coleorhiza. Now, let us have a look at this. Can you identify what this plant is? It is a germinating maize. Maize seed germinated. It is a seedling. You can see the coleoptile. Coleoptile, when the seedling comes above the soil, it comes straight, but in dicot it comes like a hook. So, the shoot tip is protected like this. It is not injured, but the monocot seedling is coming straight. The plumule would be injured. To protect the plumule, there is the coleoptile. So, coleoptile is sheath that covers the plumule, protects the seedling during germination. Now, following next is regarding the entire structure formed is seed. We discussed endosperm development followed by embryo development, then we come to seed, because seed is the ultimatum of sexual reproduction. It is the beginning or it is the ultimatum of a angiosperm and angiosperm reproduction. Seed is often described as a ripened ovule. Ovule on maturation and ripening give rise to a seed. A seed has storage structure, a seed has embryo. Embryo is surrounded by integuments. Now, we discussed what embryo is. Embryo consists of embryonal axis and the storage structure, the cotyledons or endosperm in case of monocots, but in turn it is surrounded by integuments. The integuments may be differentiated or undifferentiated as they are fused. Now, how many types of seeds are seen? We have seen the legumes. All legumes we have seen. जेते डाली जातीय गछ आमे सबु देखिछंती, सबु मंजी की देखिछंती. बादाम आमे जोटा मेजरली खाउछंती बा पर्पस, these are cotyledons. Beans, cotyledons, but thing is they are cotyledonate seeds, X-albuminous seeds. During development we have discussed two things, one is embryo, another is endosperm. Endosperm is nutritive, but embryo derives nutrition. Embryo grows at the cost of the endosperm. During development, all the endosperm is diverted to the embryo, so that in case of X-albuminous seeds, cotyledonary seeds, the storage remains in the cotyledon. That is why mungs, all the beans, they are rich with cotyledon, but others you look at, all the monocots, all the grasses, maize, rice, wheat, they are albuminous seeds, starchy seeds. the food reserve is your endosperm. That is why grass seeds are called endospermic seeds, dicot seeds are called cotyledonary seeds or X-albuminous seeds. Now, but in certain cases like your castor, it is partly endospermic and partly cotyledonary. Cotyledons are there and endosperm is there, but cotyledon is papery, it is not fleshy as it is there in the beans. Just to give you an impression regarding an unfertilized ovule, how it turns to a mature seed, these three diagrams would be very much helpful. Look at the first one. This is the egg on fertilization, zygote that has given rise to your embryo. Embryo is a contribution of both paternal and maternal parents. Endosperm is triploid. It is contributed by 2 nuclei from the female and 1 nucleus from the male, and integuments which turn to seed coat, that is purely maternal in origin. So, now it is a mosaic, seed is a mosaic structure, and in the middle one the endosperm is huge, much endosperm is there, but in the last one as the embryo grows at the cost of the endosperm, endosperm is exhausted and it goes to the embryo. Embryo has differentiated into cotyledons, its plumule and radicle, and embryo is fully developed and it is surrounded by hardly any endosperm. Now, in mature seed you will see the integuments they have fused to form the seed coat. Now, how the changes took place? Integuments turned to seed coat, zygote gave rise to matured embryo, and endosperm cell gave rise to endosperm, and subsequently endosperm is exhausted at the final stage in dicotyledonous seed. So, this is how this matured seed has diploid cell or diploid tissue, that is embryo, triploid tissue, the endosperm surrounded by diploid tissue of the integuments seed coat. Now, on completion of embryogenesis, when embryogeny is complete, then maturation takes place. What maturation? You might have seen a seed while it is in the plant, it is not having much water in it. How is it? Definitely during maturation there is desiccation, dehydration of the seed. It is seen during late embryogenesis, there is a protein called late embryogenesis abundant protein that causes dehydration of the seed. So, that the seed towards its maturity is hardly having water, 10 to 15% of water in it. So, this is about maturation of the seed. Then finally, the seed coat is derived from the integuments, it hardens, it encases the embryo and the stored food in the protective armor. You can see this any leguminous seed, you might have seen tamarind seed. The tamarind seed is well protected by the seed coat, it is difficult. You can see golmohar seed, golmohar seeds, they are also very hard because of the presence of a hard seed coat. So, seed coat prevents any damage to the underlying tissue. Now, as a result of desiccation, the metabolism of the seed comes to halt. Therefore, often it is told a seed is under suspended animation. As dehydration took place, the metabolism slows down and ultimately suspended, and in that case the seed suspends all its metabolism and comes to a resting phase and it enters the dormancy. Think of if all the cells they are falling from a plant and germinate immediately there, what will happen? So, dormancy is induced. Usually it is your abscisic acid which accumulates in the seed, which make the seed dormant. It is a chemical factor that develops within the seed. Now, have a look on this. The seed, how many seeds are there in a fruit? We have discussed about the seed. Seed is a ripened ovule, but what about the surrounding structure? The ovary, the ovary wall. The ovary wall turns into fruit wall. Therefore, a fruit contains one or many ovules. Mango has one ovule, it is one seeded fruit, but this plant, it is a leguminous plant, it has many ovules in it. Pea has many seeds in it, and why this diagram or photograph is put before you, you can see how many seeds or the seeds are attached to the placenta by means of funiculus. The funicle, the stalk of the ovule is very well marked in this diagram to understand. Now, once the seeds are matured, they are detached from the fruit wall. The point at which the funiculus was attached, that point is called hilum. You can see in this broad bean, this is Simba seeds, you can see the hilum is well marked. At that point, it was connected through funiculus to the ovary wall. Now, and near that hilum there is the microphile. Microphile you have read or you have come across. Microphile is the opening of the ovule where it is incompletely covered by integuments. So, microphile is the point in the seed which allows water, oxygen to enter and facilitates germination. Now, there are very big seeds, very small seeds. Can you see this? This is a fig. It is a ficus one. You might have seen ficus elastica, ficus religiosa, ficus bengalensis, borogacha dekhithibe. Gotte phalare so many number of tiny seeds are there. So many number of tiny seeds. Orchids, orchid plants in their capsule, they have large number of coffee sized seeds. So, the number of seeds in a fruit varies from one to many, but it depends. Now, let us come this way, how a seed or fruit is helpful. In mango we have seen which one is the seed and which one is the fruit. The stone along with the flesh, that is mesocarp, and the epidermis, epicarp, total is pericarp. So, ovary wall is totally converted to fruit wall and the inner structure is the seed. Are you interested in the seed? No. Nobody is interested in the mango seed. Therefore, angiosperms have developed fruit. The fruits are rewarding to the animals. We carry mango with us, or we are interested in mango because of the ovary part, fruit part, not the seed part, but inadvertently, unknowingly we are carrying the seeds. Therefore, in angiosperms, in some cases like your pea or all the legumes, we are never interested in the ovary wall, the fruit wall, we throw it. As it is, the seeds are proteinaceous, we take them. We go for agriculture of these leguminous crops because of the seed value, their fruit value, their protein value, but in other cases we are interested for the formation of the fruit like mango and papaya and figs and so many. Therefore, I want to say angiosperms either through their seed or formation of fruits, they help dissemination or distribution, dispersal of seeds from a parent plant to newer localities. They colonize different habitats because of certain strategies. You might have seen certain fruits, they are quite red. That red color and puppy mass attracts the birds, as a result of which they take the fruit, and ultimately unknowingly they disperse elsewhere, and it is the same case in case of banyan. Banyan fruit is rewarding to the birds. The birds eat the receptacle, the cover, the fleshy part, but unknowingly they take the seeds, and seeds are hardly digested by them. Seed is not nutritious, the fruit wall is nutritious, but for us the seeds, especially the rice, wheat, maize, other cereals are very important. We are interested in their agriculture. We have seen certain fruits in which no seed is there. For example, in certain papaya we have seen they are seedless, grapes they are seedless, your pineapple it is also seedless. So, seedless fruits is desirable. Watermelon it is very found in the summer, it is also in certain varieties it is also without seeds. So, development of seedless fruits is called parthenocarpic. So, parthenocarpic fruits lack seed as the ovules are aborted due to non fertilization, because no fertilization is there, therefore, seeds do not develop, but some chemical changes take place, as a result of which the ovary turns into fruit, and the parthenocarpy is desirable in many fruits. So, that has commercial application. Now, parthenocarpy is induced in certain flowers by application of a gibberellin, but it has not gained commercial success. Okay. Now, we will coming to this apomixes. The term itself indicates apo means without, mixes means fertilization. Everywhere there is fertilization leading to true seed formation, but apomixes is seed production without or bypassing fertilization. In absence of fertilization, there is formation of seed. If you look at, there are certain tissues, diploid tissues, if they directly develop into seed without being reduced, no reduction is there, no reduction is there, no reduction means meiosis is not there. The megaspore mother cell directly give rise to seed, unreduced gametes are formed and directly they can give rise to seed. But what is the advantage of this? When unreduced structures are there, they are genetically identical to the mother plant. So, apomixis is asexual reproduction through seeds. So, here seeds are formed, but asexually, no meiosis and no fertilization involved. What is the advantage of this? What is the advantage? It is clonal to the mother plant. Mother plant ro pura genetically identical. Apomixis has great potential for agriculture. We are acquainted with hybrid seeds. Hybrid seeds are heterozygous. They have high potential for production, or they are highly productive. Agriculturists are going for hybrid seeds cultivation, but these farmers they are not keeping their produce or their seeds for subsequent cultivation. If they keep it and cultivate in 1 or 2 or 3 seasons, the productivity decreases. The reason being the heterozygosity of the F1 hybrid is lost due to segregation during sexual reproduction. Therefore, apomixis is an alternative way by which F1 hybrid seeds can produce up to apomictic seeds. Hybrid seeds are costly, it is cost effective, highly costly, therefore, it is also troublesome. So, to avoid this, scientists are trying to produce apomictic seeds of the F1 hybrids. As a result of in future it will be a boon to the farmers. Now, around in 640 plants, this Apomixes is seen in nature. Therefore, some genes responsible for apomictic seed production are there, and researchers are interested in identifying these apomictic seeds and inducing these apomictic seeds to the cultivars like rice and wheat, so that the F1 hybrids, they can retain their heterozygosity, productivity in future generations, and that would be very useful for agriculture. In addition to Apomixes, another feature is found in certain seeds. A sexual seed, a true seed has one embryo, but citrus, all the lines, they have, in addition to this, they have from nucellus certain embryos develop, and these embryo are many in number, and that is called polyembryony. So, polyembryony is a feature along with Apomixes is encountered in angiosperms, but Apomixes would be of great value to the farmers, to the agriculture sector in the future, and research will lead its way to production of apomictic seeds. Thank you all.